Good morning, students. Hope you're doing well. We are going to, or what I'm providing here is the second lecture to chapter 18 in respect to blood. And what I'm going to do first is do a quick review of the last lecture, and then I'll start with the second part of this lecture. There could actually be a third lecture with this. I might break it up and do blood typing and talk about how platelets are formed, and then I might make a different section. But anyway, you'll see that posting. So I'm also going to take my video away so you don't see me in the corner and it's not distracting to you for showing the notes. So I'm going to share the screen and show the first set of notes that we've taken. Hopefully you're following along and taking these notes. So, okay, let's go. Share screen. Oh, let's see. Sorry, I was going to, yes, I can do this. I was going to stop my video so that it's not distracting. I don't know if my name is up there still or not, but anyway, moving on. Actually, before I actually go into the notes, I did want to show you this slide. This slide happens to be a blood smear, and how they make that is they put a drop of blood on a microscope slide and then take a cover slip, which is just a smaller piece of glass square, and they'd smear it along to just make a flat surface of the blood to be able to view it under the microscope. And if you were to do this, you're going to see all of these light pink looking cells. These are red blood cells, and also known as erythrocytes. They have lost their nucleus in their formation to become a mature erythrocyte. And so you can see it's kind of really thin in the middle where the nucleus used to be. It's not a complete donut. There's not a hole there, but and this will allow it to carry oxygen to your brain, to your body parts, and also to pick up carbon dioxide and get rid of it through your lungs. These larger cells you see, these are the white blood cells. And as we established already, there's 5 types of white blood cells. There's an acronym I told you to help remember is BEN, B-E-N. So basophils, eosinophils, and neutrophils. And then there's also monocytes and lymphocytes. And monocytes and lymphocytes give rise to other cells, white blood cells, that we'll learn more when we talk about the immune system. Over here, the erythrocytes have been stained blue, just for you to see it. Down here, the neutrophils are stained blue in this case, and the erythrocytes are their normal color. And down here, the little blue fragments you see in this lower slide is of platelets. And remember, these are just fragments of cells, and they happen to be fragments of megakaryocytes. And megakaryocytes were given rise from monocytes, which happens to be one of the 5 white blood cells that we're studying now. So anyway, I just wanted to show this slide to you. Now, going back, I'm going to stop sharing this and share our notes. Okay, so for a quick review, chapter 18, we talked about the blood. The circulatory system consists of the heart, the blood vessels, and the blood itself. The cardiovascular system consists of the heart and blood vessels that we'll study in upcoming chapters. TPR, blood helps with transport, protection, and regulation. Primarily transport is oxygen and carbon dioxide, but we also have metabolic waste that we want to get rid of. We want to take the nutrients from the stomach to other parts of the body. It carries hormones. Protection, it brings the white blood cells to the site of injury. And the regulation, it helps stabilize fluid and fluid distribution, buffers acids and bases so that we can keep your blood at 7.35 to 7.45. We don't want it to fall out of this pH range. I asked you to please review table 18.1, the general properties of blood. Adults have 4 to 6 liters of blood, and it is considered a type of connective tissue. Here, talking about the formed elements, there's 7. We don't call them cells because platelets are not cells. These are fragments of cells, and specifically megakaryocytes. Okay. We'll talk more about that later. The erythrocytes are the red blood cells, and leukocytes, there's 5 types as discussed: baso, cyno, neutrophils, lymphocytes, and monocytes. They can be either granulated or agranulated. So they're granulocytes means that you can see the proteins in the cytoplasm. If they're agranulated or agranulocytes, the proteins are not as visible. They're still there, but they're just not. And these are proteins to help fight bacteria. We also discussed that if you put blood in a centrifuge, the heavier particles will go to the bottom. 45% happens to be the rest, makes up erythrocytes of whole blood. Plasma makes up 55%, and then there's like it looks like a layer of snot on the side of, in the middle of a test tube, and this is the buffy coat, which contains the leukocytes and platelets. I gave you some information about numbers here, and then I discussed plasma and what you would find in plasma. Make sure you can review that. I talked about specifically then erythrocytes. Erythrocytes, they have glycoproteins on their outside, on their membrane, and these are antigens, and antigens are what we'll use to talk about blood typing. Erythrocytes, the 2 main functions: to pick up oxygen and pick up carbon dioxide and take it where it needs to go. Lifespan, 120 days. Has a biconcave structure. Why? Because at one time there was a nucleus in here, but it was ejected. All right. Hemoglobin, that's what makes up 97% of the red blood cells of a red blood cell. There's made up of 4 proteins, globular proteins, and we discussed globular proteins when we talked about plasma. You got 2 alpha, 2 beta. The red is representing the iron, and then each iron contains an oxygen. So that right there would be like an oxygen. So this is just one molecule of hemoglobin. And a red blood cells has plenty of hemoglobin, lots of hemoglobin. There is a number, and I can't recall it right now, but it's like millions. So one hemoglobin can carry 4 oxygen molecules, and you have to have an iron in order for the oxygen to connect. Carbon dioxide, so if you unload oxygen, then the red blood cell wants to pick up carbon dioxide, and it does not adhere to the iron. It adheres to the globular proteins. We'll talk about that further later on as well. Then we discuss the formation of erythrocytes, and we know that it comes from stem cells that's found in the ends of the bones. So this is called hematopoietic stem cells. And so in here you have a lot of little red blood cells, okay? These are immature, and they're not called erythrocytes, they're called erythrocyte colony forming units. On each of these cells you have receptors to receive a very important protein, well, it is a protein, but a hormone called erythropoietin, EPO. And this is produced by the kidneys. And once the hormone, if we're going to represent the hormone in green, it will hook on to the receptors, all right? And this causes, EPO causes the ECFU, the erythrocyte colony forming units, to turn into erythroblast. And these cells will increase in number, and they also synthesize hemoglobin, by the ribosomes. So the cells are proliferating and making lots of ribosomes to make a lot of hemoglobin. The next stage, the nucleus will be discharged, and now we call it a reticulocyte. This is an immature erythrocyte. However, as it starts to leave the bone, so it's going to, the cell's going to leave the bone, it's going to enter into the blood system. And once it enters, then it gets rid of the rest of the ribosomes because we have enough hemoglobin made for that cell. And now it's referred to as a mature erythrocyte. Okay, so once it leaves the bone and gets into the circulatory system, now it's mature. So if I were to ask you this on the SOE question, please describe the production of a red blood cell, then you would tell me the 4 main parts of what's going on, and then the details with each stage that's going on to finally get to mature erythrocyte. Now, one more thing I added about this is the hypoxia-inducible factor. This is produced from the kidneys, and it detects oxygen levels, all right? So if oxygen levels are really high, then the oxygen actually destroys this stuff, okay? It destroys it. But if oxygen levels are down, the kidneys will produce HIF, and when you have production of HIF, then you will produce more EPO. So low oxygen increases HIF. Sorry, I left the I out. And then therefore, the 3 dots represents therefore you increase the EPO. And the more EPO you have, the more erythroblasts you can make. All right, so therefore you're increasing your red blood cell. So we have to monitor oxygen levels, and what happens is this hypoxia-inducing factor is one that's monitoring the release of EPO. So if there's lots of oxygen, you destroy the HIF. If there's low oxygen, then HIF will be produced from the kidneys, and then it will create more EPO. I think that's where I left you. Now we're starting in brand new material, and with this new material, we're going to have an understanding of blood typing. Okay. So, like I said just a minute ago, on your red blood cells, there's a red blood cell I've just drawn. You have carbohydrate or glycoproteins. Okay. Some are spiked shape. Some may be like a block. Now, this is not going to make sense right off the bat, but just stay with me. All right. So, depending on your red blood cell, it depends on what type of antigen you have on your red blood cell determines your blood type. Okay. And there's lots of blood types. There's A, B, C, D, E, F, G, but ABO happens to be the one you're familiar with and is the most crucial in determining blood type. So, and there's another one, D, but we'll talk about that in 1 minute. So blood typing is determined by the type of antigen on the red blood cell. Erythrocyte is the same meaning as red blood cell. RBC is easier for me to write. An antigen is a glycoprotein or a glycolipid, and I'll just take this and point to it. Right there's the glycoprotein, okay? And that's also a glycoprotein. So these are antigens. The APO antigens, hopefully I'm recording. I'm sure I am. Determine the blood type. All right. So in this case, this one I'm going to have, these are just symbols to represent. This one, it looks like a triangle. I'm going to call this antigen A. all right? The block, I'm going to refer to that as antigen B. And now these structures over here, these are antibodies, okay? And in this case, the one that looks like an upside down Y, I'm going to call it antibody A. And this one is antibody, or antibody, I shouldn't put, okay, B. Now I'm going to go to scratch time for a minute and try to clear up some stuff before I go here, okay? So if you have a red blood cell, all right, I should make it red. This is just scratch to help explain. And I have this triangle on here, and no blocks, then this is going to be blood type A, because we just designated the triangle as antigen A. So there are antigens on there. Now, in your blood, let's just take a glass of blood, and let's say your blood type A, and we know that. I'm going to put your cell in here. All right. Well, you know, you also have plasma, and in the plasma, we've already established that you have antibodies. So these antibodies, if in A you had antibody A, you can see that this would fit right here, okay? This is bad, bad. This causes agglutination, right? Clumping of the blood, that's what that means, clumping. And this can lead to death, blood clots, aneurysms, okay? So in blood type, if you have, this is your blood, you do not have the A antigen. What you have is you have the A antigen. You don't have the A antibody. What you do have is the B antibody, also known as anti-B, okay? So this, if you can see, if your red blood cells have the triangles, this will not fit on there, all right? So it's fine. It's perfect. It's good to have these in your body, no problem, all right? So if somebody, look here, I'm putting a block. I know we designated that to represent the B antigen. So this is blood type B. Let's put their blood in a jar. We have the block. So my question to you would be, what kind of antibodies would you find in here? Would you find antibodies A or antibodies B? If you found antibodies B, and we know what it looks like, it's a block-shaped antibody. If that was floating in here, all right, that was in there, that would attach to the red blood cell. And once again, if this went here, this is bad, okay? Once again, it causes agglutination. So if I were to ask you what kind of antibodies would you find in blood type B, the type you would find would be anti-A. So this is anti-A. This is blood type B. So these are compatible, all right? Why? Because this will not cause clumping. The antibodies will not stick on all of the little antigens of type B. So hopefully this makes sense to you guys, all right? If not, always email me questions. I will Zoom with you and I will talk with you pretty much anytime to help further explain this. You just gotta ask for help, so I'll know. All right, now let's throw something else out there. Let's say you have a red blood cell, okay? There's no antigens. Nothing stuck to the membrane of the red blood cell, no antigens. So this is called blood type O. There's no antigens, all right? So now I'm gonna put this blood in a beaker. So we've got our red blood cells, all right? We know they're O, type O. My question to you, what kind of antibodies can you find in here? Would it be okay to find an antibody that looked like this? would it be okay to find an antibody that looks like this? Remember, this is anti-A, this is anti-B, this is type O blood. Type O, nothing on it, all right? So since there's nothing on here, there's not any antigens, okay, at least ABO. So therefore, these antibodies can be in this blood, okay? This is all good. Great. So now let's look back at our chart because I'm gonna build on this, okay? So you have to have an understanding of the foundation because I'm gonna build on it. So here, you've got your symbols to represent what you're looking at. And think of it as a puzzle. So in this, I can see the red blood cell and I can see that it has the A-shaped antigen. So I know that that blood type is A. What kind of antigens do I see there? Of course, A antigens. What kind of antibodies do I see in here? I'll look at the block-shaped antibodies. I'll look over here and I see anti-B. Okay, let's go to the next one. I see blocks, so I know that it's type B. I know that the antigens involved, it's B, all right. And I see in here, upside down Ys, these are the antibodies A. So that's fine, they can be in there, okay? You would not want antibodies B in here because you'd have clumping. So in this case, we have the antibodies found in this blood, anti-A. This one, okay, let's say that there's no antigens. So I know that the blood type is O. Are there any antigens? None. Okay, so what kind of antibodies can be in here? And as we already expressed, we could have B and we could have A. Since there's nothing for it to stick onto, that's great. So antibodies can include anti-A and anti-B. All right, now I'm going to throw you a curveball. Let's say, where's my nice blue? I'm putting blocks on this red blood cell. Here's another block. And I'm going to add spikes, all right? And I'm going to ask these questions. What blood type? Well, since I have A and B antigens, this is going to be blood type AB. Antigens involved are A and antigen B. Antibodies available? None. Because think about it. If I had antibodies in here, then they would stick and you would have clumping. So that is the basics of blood typing. All right. Now, so blood type O here, no, blood type O is the universal donor. I'm going to explain this in a minute. All right. Type AB blood is known as the universal recipient. Now let's think about donating blood. I'm going to have to go back to scratch paper to show you and explain this. So got some scratch paper here. Now, who can give blood to whom? All right. I'll get my pen. Let's see. All right. So let's take this person right here. All right. Okay. Here's their blood in a beaker. All right. And I know that their blood type has a spike. So what blood type does that have to be? That has to be blood type A. All right. What antibodies are in here? Has to be anti-B. Okay. So this is anti-B. Draw another person. Okay. Put their blood in here. All right. So once again, I'm going to put blots on this. So I know this is the antigen B, blood type B. Okay. If you forget, you can refer to your little chart here. They, have antigens that look not like blocks because they would have clumping. They look like upside down Ys. This is anti-A. Now, when you donate blood, you can either donate the red blood cells or the plasma, okay, depending on what the person needs. So if this is the recipient, they were in Iraq and they need red blood cells, could they take this person's red blood cells? Could they take these red blood cells? Now remember, the plasma stays back, so this is not going. The anti-B is not going, all right? Because right now we're just talking about the red blood cells. Are the red blood cells coming over? Yes. So could this type B receive from type A? Well, think about it. If we put this cell in here, right here, we put this cell in here, it has a spike on it. This antibody is going to come over and attack it because it thinks it's a foreign invader. See? Okay? So, no. Type B blood, or somebody with type B, can not receive RBCs from type A blood. All right, so this cannot happen. All right, you were like, well, could you give the plasma to the person? Well, here's the plasma and it has this anti-B in it. So let's see, let's put it in here. No, because what that would do, that would clump right here to here, and so there. So a person cannot take type B blood, all right? And a type A person cannot take type B blood. So this person has been in a wreck. They need somebody's blood, all right? They need somebody's red blood cells. So if we have another person here and says, hey, can I give some blood? Well, I don't know. Let's look at the antigen on your red blood cells. Remember, this person has antibody A. So if you have a red blood cell, okay, and it has blocked antigen, so this is type B, this blood looks just like this blood, so these red blood cells, yes, type B can donate to type B. Could this one come in here? No, because that A antibody anti-B would clump onto this, and so no, this could not give to him. But this, yes, this blood could give to this, right? Is there anybody else that could give to this blood? Well, let's try this person here. Let's look at their blood in a beaker. And there's no antigens, nothing. So this is type O. And now let's take this red blood cell and put it in with this person. Will this connect to that? No. All right. So type O could give to type B, and type O could give to type A. Now, for me, I always have to draw out these pictures and think about it and look at the antigens and the antibodies to see whether it's a match or not, okay? Since there's no antigens on here, that's great, okay? And this blood, remember, you're only donating red blood cells, okay? Now, in the plasma of type O, you could have these, because this person, there's no antigen here for that to clump on. So yes, those could be present in the plasma. That's the key word there. Oh gosh. That's supposed to be a block. Okay. It can have anti-A, anti-B. Do a better anti-B. Do another anti-A. Okay. So you would not want to give the plasma from type O to type B, because these antibodies, like, well, this one right here, this antibody would attach here and cause clumping. But remember, when you donate blood, you either donate the red blood cells or you donate the plasma. You have to just look, and if there's a connection between the puzzle pieces, you're going to have clumping, and that donation cannot happen. But since this is a clean red blood cell, no antigens on it, then that's why type O blood can donate red blood cells to anybody. Therefore, it's known as the universal. Type O blood can donate red blood cells to anybody, and therefore it's known as the universal donor. Okay. So let's go back to our chart here, to your organized notes. I've got a lot of scratch paper here. And so hopefully that gives you an understanding of why type O blood is the universal donor, because there's not any antigens on the red blood cell. Type AB blood is the universal recipient, okay? Because, let's just look at this and make sure I'm clear about this. So here, I'm going to take somebody with type AB blood, all right? There's the B antigen, there's the A antigen, B antigen. So this is a type AB blood, all right? This is known as the universal recipient, all right? So if you think of all the other blood types, let's just do them real quickly here. There's the spike. I'll put an A there for type A. And this is type B, the block. And this is O, no antigen, all right? So think about it. This blood in the plasma, put a little note here, the plasma does not contain anti-A or anti-B. So if these red blood cells come over here, all right, let's put it here. There's a spike. There's not any antibodies A to clump on here, because if there were, it would clump to its own blood, and that would be bad. So yes, these red blood cells can come over. Can this come over? Yes. Okay, so therefore AB can receive blood from all types. I'm talking about, this is where students make their mistake. They're like, well, look, this one has anti-A in it. If you bring it over here, then you're going to have clumping on here, right? Well, remember, you're only donating the red blood cell, not the plasma, okay? So if you're talking about plasma, you're right. The plasma cannot come over here because it would clump, okay? But we're talking about the red blood cells. So yes, that could be, that's all positive, that could happen. So therefore, this should explain why type AB blood is the universal recipient. Now, let's throw in one more thing, okay? Let's see. Yes. They found, you've heard of blood being called A positive or B positive or AB negative or A negative, what have you. Well, they discovered, and it happened to be on rhesus monkeys, the rhesus antigen. So this is named after the rhesus, it's a type of monkey, and it was one more antigen they found that was very important, and either you have it or you don't have it, okay? And the way I'm going to represent this antigen is is with D. So right here, the RH equals, I'm going to have it look like a half moon, half dome shape. The RH antigen, this is the antigen, is also referred to as the D antigen. So A, B, D. They skip C. It's supposed to be RH right there. Put a little white out so it looks more like an H. All right, my white out is about shot. So the RH antigen, I'm going to represent it with the dome shape, and this is also referred to as the D antigen. The D antigen equals the RH antigen. They can be used interchangeably. Usually they say, what is the RH factor? Is it positive or negative? That's the question. All right, so I'm going to have a red blood cell right here. And I see that I have a little plus in here. So that means it's positive. So what does that mean? That means that the antigen is present on the outside of the cell membrane. Okay. And in this case, there's none. So it's RH negative. So let's look at this for a minute. So here's a cell. It has a dome-shaped antigen on it. Okay. And it happens to have also a block on it. So from what you've learned, if I ask you the blood type of this is blood type B, has the RH factor on it, so it's B positive. Okay, that's B positive. Look at this blood. It has the antigen. Doesn't have any other antigens, so this is blood type O positive. Okay, so now you have to look at the positive and negative as well to make sure whether it can receive blood or not. So let's just take an example. Let's take some blood. I'm going to put, I'm just going to put O positive in it. So there's type O positive. Let me put my antigen on here. Okay. So my question to you is, what antibodies could I find in the plasma? Well, I could find A and I could find B because this doesn't have A or B antigens. But would I find antibody, not antigen, antibody D that looks like this? It's arched. Would I find that in that blood? The answer is no. You would not find this antigen in this blood. Okay, I said antigen, I meant antibody. All right, so let's say this, we know the blood type O is the universal donor, yes? Yes. So let's take another beaker of blood, okay? And in this beaker, let's say in the plasma, I have these antibodies. And let's say that I have this antibody, okay? So what does the red blood cell look like? Well, it could look like O, but I'm going to just put this on, yeah, let's just say that it just looks like this, all right? So my question to you is, What type of blood is this? Well, there's no antigens on here. None. So it's type O. It has antibody D in the plasma. So therefore, it does not have the half moon on it. If it did, you would have clumping. So if we had the D antigen on here, this would clump onto that. So that's not there. So let's look at this. Could this blood, let's just call this beaker number 2 and beaker number 1. Could beaker number 2 donate to beaker number 1? And could it donate red blood cells? So the question is, could beaker number 2 donate to beaker number 1? And when I say donate, I mean the red blood cells. So I'll just put this up here. Could beaker number 2 donate red blood cells to beaker number 1? All right, let's just draw it and see. So let's take, this is beaker number 2. Let's take this red blood cell. We're going to donate some red blood cells to beaker number 1. Let's put it in here. All right. All right. There's not anything. This would not fit on there. This would not fit on there. So the answer is yes. You could donate just fine the red blood cells. Now, would you want to put the plasma in here? No, because these would go and attach right there. So no, right? Now let's change this around. Could beaker number 1 donate red blood cells to beaker, how do you spell beaker? Gosh, I misspelled it a lot, sorry. To beaker, could beaker number 1 donate red blood cells to beaker number 2? Well, let's just try it, all right? So here's the red blood cell. I'm gonna put it over here. Right here's the red blood cell, type O, and I'm put the antigen on here, okay, because it has the antigen. And look, this antibody would come right down here and cause clumping. So therefore, no, you cannot go this way, all right, because you would have clumping. This way, yes. Everybody's happy, okay? Now remember, we're talking about the red blood cells, not the plasma. So hopefully that makes sense, all right? So you have to remember that Rh positive and Rh negative. Now there's a disorder that you need to be aware of, and that is what would happen if a Rh mother was pregnant with an Rh positive fetus? Okay, and I've got some drawings here, but let's just draw that question out. And when you have the essay questions, this is what you need to do. You need to draw everything out, okay? We are only looking at the Rh factor, all right? So what would happen if an Rh mother, so here's a mother, well, this is terrible drawing, so, but let's just say we have a mother, and she's pregnant, all right, and in her blood, she is Rh negative. So she has red blood cells, and it's Rh negative. Okay. So what would happen if the Rh mother was pregnant with an Rh positive fetus? Now here in the fetus, we have Junior, okay? And his blood happens to be positive. Just drawing it big so you can see, okay? So the fetus blood supply is separated from the mother's blood supply. So for the first pregnancy, she could probably have this baby and there's no problem. There's absolutely no problem, all right? However, if there happened to be mixing of blood, either during the delivery, there was, this tearing going on during delivery, or if it's cesarean or anything, if some of that baby's blood were to get into the mother's blood, okay, so let's say I'm going to represent the baby, and that baby's blood happens to have the D antigen on it, this mother will start making antibodies. Okay? So now her immune system has started making antibodies to attack this foreign blood that got in here during delivery. Okay? All right, now let's have, now she's going to get pregnant again. Okay, so we've got, this is the second pregnancy. All right, so we have her represented, and then we have the baby. Remember, she is Rh negative. All right, however, in her plasma now, she has these guys floating around. Why? Because her immune system, when this came over during the pregnancy, during delivery, the immune system made antibodies against it. So if these antibodies happen to get in with the fetus here, and this fetus, once again, is Rh positive, and these antibodies travel over here, if they slip through, then it's going to attach. It's going to cause problems, and it actually leads. So we're going to have clumping, and it causes a disorder that is referred to as the hemolytic disease. Or it's also known as erythroblastosis fetalis. So what happens is, we need to prevent this, okay? So what happens is, to prevent this, in the very first pregnancy, they're going to determine the blood type of the fetus. And they're going to give what is called a RhoGAM. To prevent the disorder right here, to prevent hemolytic disease, what's called as a RhoGAM shot is given at the 28th week of gestation, and 72 hours before delivery. Okay, so what happens is, if this shot is given, so let me just draw the mother here. This is the first pregnancy. Okay, Junior is Rh positive. Okay, the mother is Rh negative. She's no D antigens, no Rh antigen on here. So they give it at the 28th week and 72 hours before delivery. So just in case, so they give a RhoGAM shot. And what is this? This happens to be D antibodies. okay? Not made from her immune system. The immune system is not making them. It's just enough so that if the baby's blood comes over here during delivery or there's mixing of the blood one way or the other, then what happens is this antibody will clump onto here, okay, causing the mother's immune system not to make its own antibodies. Therefore, future pregnancies are protected. And this one. Okay? And what happens, hemolytic, if something is lytic, that means that you're destroying the erythrocytes in this case, and therefore the infant can be anemic. And if it's anemic, if something is anemic, it's low oxygen content. So actually what happens is the infant will, if its red blood cells are being attacked, the infant will increase EPO secretion, and it's actually making erythrocytes that are immature. And then this leads to all kinds of problems. It can lead to jaundice, lots of red blood cells that are being broken down because they're not mature, and a lot of the treatment will be UV light to help take care of all these red blood cells that's being broken down. So anyway, that's a little bit about that. I'm sure you can Google and learn more about this disorder, but this is the gist of it. So right here, back to our formal notes. What would happen if RH mother answered? If fetal blood crosses the maternal blood, mother will make antibodies against the fetus blood that causes, like I said, erythro, make sure I spell it right, blastosis fetalis, also known as the hemolytic disease. Okay. As explained, and this is a RhoGAM shot. It's a shot of D antibodies. So if you put them in artificially, it will not spark the mother's immune system to become sensitized to her fetus. All right, so I am going to pause this recording for a minute. All right, students, hope you took a little break. I did. I just want to, hopefully you're seeing this PowerPoint on leukocytes. And as I stated earlier, there are 5 different types of leukocytes. And they're also based on 3 of them are considered to be granulocytes and 2 of them agranulocytes. So the ones that are granulocytes contain granules, and these are proteins called defensins that fight bacteria. And all white blood cells have these granules. It's just that they're obvious in 3 types of the white blood cells. This would include granulocytes, would include the neutrophils, eosinophils, and basophils, BEN. B-E-N, to help you remember. And agranulocytes would be the lymphocytes and the monocytes. I'm going to talk about these individually with our outline notes, and obviously you can see it's in the PowerPoint too. Here are the granulocytes. Notice that this purple lobe, this has like several lobes in it. These are the neutrophils, and they're primarily responsible for fighting bacterial infections. Here you see it's bilobed. There is a little connection between here, this nucleus, and this is the eosinophils. And then the basophils is a very large nucleus in the whole thing. The granules are these little purple dots stained here. Here the granules are pink, and here they're kind of, they're purple, okay? Once again, this is a neutrophil. It's multi-lobed nucleus. The granulocytes stained kind of purplish, cross between pink and blue. The little red things around it are the erythrocytes, the red blood cells. The eosinophils, it's not multi-lobed, but it's bilobed, and it's pretty big. And just different views of the eosinophil. And basophils, the nucleus is rather large. And then I don't have the agranulocytes. I don't think I have a good picture of those, but I'm pretty sure it was in your reading. So I'm going to stop sharing this and go to our formal notes. Here we go. And let's go over some of this information. So basically, the leukocytes, these are cells that provide protection against infection and diseases. They only spend a few hours in the bloodstream, mainly they use the bloodstream as a subway to get to the damaged area where there's a pathogen coming in. A pathogen is, anything that can cause a disease. They retain all their organelles, unlike red blood cells who eject the nucleus and get rid of their ribosomes. They keep all their organelles for life. And to distinguish between the different types of leukocytes, what they look for, well, if there's cytoplasm granules, if they're present or not. Okay, whether they're present or not present, whether they can be stained or not. Size of the cell. We also look at shape of the nuclei. And the I'm going to put staining properties. Well, staining properties right here, because the staining will show if they have granules or not. And then, of course, the function. There's three main ones. And the way I actually meant to put BEN this way, but the way I drew it, these are the neutrophils. These are the most abundant. And then we have the basophils. And that leaves us with the eosinophils. Okay. The staining in this case, you use a right stain. To be able to see, and this one is kind of purplish when it stains. So you can see the granules in here. So the neutrophils, you have increased neutrophils for a bacterial infection. Pus equals dead neutrophils. So if you get a cut and it gets infected, you see a lot of ooze of this yellowish gunk, that's pus, and that is the dead neutrophils. And then these are the most abundant of the white blood cell count. And they make up about 60 to 70% of all white blood cells. Okay, the basophils, these stain blue with the right string. All of these, keep in mind, are granulocytes because they have the granules you can see stained. And this one's very important. It secretes two chemicals, histamine and histamine causes vasodilation. All right. Therefore, neutrophils and clotting proteins can get to the infection or injury. Okay, so it makes it dilated so these other white blood cells can get there to help fight the problem. Well, you might have heard of antihistamines, or during the time when there's a lot of allergens. Your body, if you breathe in allergens, pollen, the body's going to think that this is foreign, and you're going to react to it. So it's really a hypersensitive immune system that reacts to pollen and can cause runny nose and itchy eyes. It's because you have an abundance of this chemical right here, histamine, that is causing the effects that are making you feel bad. So when you take Benadryl, it's an antihistamine, so it will bind to this histamine and hopefully not trigger an inflammatory response. Another one is heparin. So they both start with H's, histamine and heparin. This is an anticoagulant. What that means is it keeps the blood from clotting. Once again, white blood cells, since the blood's not clotting, white blood cells can get to the site of injury or infection. Oops. Okay. And these are not very many, these are not really plentiful. It's like less than 0.5% of the white blood cell count. Okay. Eosinophils, they stain pinkish. And these increase in number when infected with parasites. Worms. Remember the Hobbit? And I think it's the Hobbit. He's like, I got worms in my tubes. Increase in number with parasitic infection. They also can increase in number as well with allergies. So if you take a white blood cell count and you see that somebody's really high in eosinophils, there's a good chance that maybe they have worms. And they make up 2 to 4% of the white blood cell count. So these are the granulocytes. Now let's talk about the agranulocytes. There's two types. The lymphocytes. They're second in they have a huge, large nucleus. They make up 25 to 33% of the white blood cell count. So second in abundance to the neutrophils. And well, the function of these is destroy cancer cells and or viral infections, excuse me. They present antigens. We should know a little bit about antigens now. These also have antigens, but it's not important in blood typing. These antigens present antigens to activate other cells. Now, these antigens, so lymphocytes can be what's referred to as antigen presenting cells. And they're also APCs. And when we study the immune system, this will come back to us. So I'll tell you more about that in just a few more minutes. These secrete antibodies to fight infection. And it also provides immune memory. So let's say you have a cold, a cold, which is normally caused by a virus, and you're sick, you feel wretched, and you recover. Well, if you come in contact with that virus again, your body should mount an immune response quickly and tackle the pathogen quickly, and you don't even know you're sick. So that's why there's so much surprise that there's a handful of people that they say have been infected with coronavirus 2 times. Well, if you're infected for the first time, technically, or what we know, you really should be immune from it. But if the virus changes its shape a little bit and comes back into your body, since it's a little different shape, you might get sick again. So I just don't think they know quite enough about it to know whether you're infected again with the same shape coronavirus or if it has mutated a bit. So it provides immune memory. You know, for example, you might get a tetanus shot, and that's giving you a little bit of the attenuated virus. It's a weakened or deadened virus to make you just a little bit sick so that your immune system can say, oh, that's what it looks like. If I see the real thing come into the body, I can mount a response and kill it. So, and then you have to have boosters because you want to remind your immune system, hey, don't forget this was a bad pathogen. So the lymphocytes are responsible for providing immune memory, and then this can last weeks to decades. Okay, so like your smallpox shot or your polio, those can last about a lifetime. So monocytes, it's another branch, another agranulocyte. Monocytes are usually the largest white blood cell. It makes up about 3 to 8% of the white blood cell count. These increase in number, up arrow means increase, with inflammation and viral infection as well. Okay, one type of monocyte is called a macrophage, a macrophage. And what this does is it consumes, a macrophage is a type of monocyte that consumes dead, dying host cells, foreign cells, red blood cells that have died, chews them up, and it consumes 25% of their own volume every hour. So they eat all the time. They don't eat, well, they destroy things and break it down. For example, red blood cells, they break down the membrane so that those amino acids can be reused for whatever. This macrophage, so is considered an antigen presenting cell. So if this were a macrophage right here, okay, and it phagocytized, meaning it took in an object, let's say something bad, like I just drew a little bug. It may present part of the bug's leg to other cells to spark a response. So this is considered an antigen presenting cell. And there's 3 or 4 of these antigen presenting cells, and I'll tell you about them as long as they go. But basically it's a cell that will present a fragment of whatever it's breaking down, so that it's like an alarm to other cells to start thinking of doing an immune response for whatever the pathogen that this macrophage might have consumed. That's just a little bit basic. You can read the PowerPoint as well about the leukocytes. To discuss how leukocytes are made, I don't go in detail like I did with the red blood cells with this one, but leukopoiesis is the production of white blood cells, and it starts in hemopoietic stem cells, just like the other ones. So, you know, in the end bones, you have this special tissue, stem cell tissue. And this is where we get our red blood cells from here, as well as our white blood cells from here, okay? So leukocytes, just like the erythrocyte colony forming unit, we have colony forming units, leukocyte colony forming units. And what this, let me get my different pen. This contains receptors for stuff that's like EPO to stimulate it. So, but there's not a specific name. So, but just know that it contains receptors for colony stimulating factors. And these factors are actually, they come from neutrophils and macrophages. Depending on the type of infection, determines the type of CFU. So if you have worms, you're going to want to make more eosinophils, what have you. So it depends on the type of infection, leukemia, or depends on what you're going to produce. So the precursor, so just like the reticulocyte was an immature erythrocyte, you've got immature white blood cells. So, and what cells give rise to the cells that we've already discussed would be, so you've got your myeloblast, kind of like an erythroblast tissue, and that gives rise to BEN, B-E-N, basophils, eosinophils, and neutrophils. Basophils, I'll just put baso, cyno, and neutrophils. Okay. You can get monoblast. Let's do it another color. And of course, these would give rise to monocytes. And the type of monocyte is a macrophage. And then we have lymphoblasts. And what do they give rise to? Lymphocytes. Okay, so those are 5 types of white blood cells: bin, monocytes, and lymphocytes. So that's just a short version, very short, of how they are produced. Couple of leukocyte disorders. Leukopenia is when white blood cell count is below normal. What is normal? Normal is anywhere between 5,000 to 10,000 white blood cells per microliter. Okay, so that would be normal. So if you have below 5,000, it's called leukopenia. What can causes this? Anything basically that kind of depresses the immune system, or poisoning, lead, arsenic, poisoning, radiation can suppress your immune system, your white blood cell count, certain viruses, HIV, such as polio. So if there's a low count, they're going to look for these type of disorders. Chemotherapy can also cause a depression in the white blood cell count. Leukocytosis is the opposite. This is when the white blood cell count is greater than 10,000. And sometimes emotional stress can cause this. Let's see, what do I need to say here? Let's just say it's due to allergies can cause it. Dehydration. Infections of any sort. Emotional stress. That can go both ways, because with stress, as we said, cortisol is released, which can suppress your white blood cell count too. What do we need here? Okay, so lymphoid, there's a couple of types of leukemia. Leukemia is cancer of the hematopoietic stem cells. And it produces too many white blood cells, and they're immature. Okay. The normal range, that's what I meant to put here, is 5,000 to 10,000. So if it's above 10,000, greater than 10,000, then there's a problem. There's 2 types of leukemia: myeloid leukemia, and this is uncontrolled production of myeloblasts. So up here, the myeloblasts that we talked about, it's when you have uncontrolled. And lymphoid is, just like it sounds, uncontrolled production of lymphocytes. Okay, both of these can be acute. So both of these can be acute or chronic. Usually if it's chronic, you have about 3 years to live. If it's acute, you have about 3 months. Okay, so it just depends on the type. Lymphoblast leukemia, this is the most common type in children, but the best thing is the most treatable, which is good, and treatable with chemo and bone marrow transplants from a match. Obvious leukemia, it's not obvious, but leukemia causes increased white blood cells that are immature. Therefore, they can't protect you. Sometimes they call it the army of children, which is referring to immature white blood cells. Oh, good. Yes. All right. So that's a little bit about white blood cells. That's good. So we've talked about red blood cells. We've talked about white blood cells. We've talked about blood typing, how the cells are formed. Now, I just want to touch a little bit on hemostasis and the production of platelets. How are they produced? And it's kind of neat. So hemostasis means, what it means is stopping blood flow or cessation. I'm going to put stopping here just to make sure you know what that means. Cessation of bleeding. And platelets are crucial for this to take place. Platelets are fragments of bone marrow cells. Okay. Which specific bone marrow cells are we talking about? We are talking about, just put called megakaryocytes. And these come from monocytes. All right. Thrombopoiesis is production of platelets. How? How does this happen? Okay. So this is produced in the liver and the kidneys. What is produced in the liver and kidneys? Is a hormone called thrombopoietin. Okay, so this hormone called, okay, I'm sorry, I'm losing my train of thought. Okay, thrombopoiesis, the production of platelets, is produced in the liver and kidneys in, a hormone is produced in liver and kidneys called thrombopoietin. And there's certain receptors on monocytes for thrombopoietin. I have to write that word again. Okay, so basically these monocytes then will, if they have these receptors for thrombopoietin, will turn into megakaryocytes. So let's look over here at the drawing. So this is a megakaryocyte. And why does it become mega? Well, what happens is this happens because the DNA duplicates However, let's mark that out, without cell division. So you know how the genetic information is duplicated and then the cell splits, like in mitosis? Well, we're not having cell division, so this cell gets huge, all right? So now we have this huge cell, and where is this huge cell? Well, we have a bone. We have a bone, and inside of it, we have this cell that had some of the monocytes had certain receptors for thrombopoietin. This cell just gets larger and larger and larger, so large that it cannot slip through and get into the, what in the world? Why is that upside down? I just looked at the screen. Has it been upside down the whole time? Oh, Lord, help me. Okay, I completely apologize. When I looked at the screen to see what I was recording, I could see that it was upside down. And I am not sure when that took place, but I want to continue with this lecture because I'm so far in and I don't have time to do it over before posting it. Today's Tuesday. Well, I might. It's not posting it until Thursday, but anyway, apologize about that. So what I was saying was we have hemostasis, cessation of bleeding. Platelets are fragments of megakaryocytes. How do these cells get so big from a monocyte? Is the DNA duplicates without cell division. So right here, I was drawing a megakaryocyte and it cannot get to the blood supply. So what it does, it kind of sends out, kind of like the blob, it sends out its tendrils. And as the tendrils get into the blood supply, the flow of the blood causes the platelets to actually break off these tendrils. So this is why platelets are fragments of a megakaryocyte. So the tendrils squeak out, get into the blood supply, and then the fragments break off. And these platelets are responsible for causing blood clotting. We're going to look at blood clotting in a minute. Function of platelets. They actually cause vasoconstriction. That makes sense. You want to vasoconstrict the area to stop the blood from coming out. They create a, sorry, I'm looking for my pen. I lost it. They create platelet plugs. Platelet plugs, which are sticky. They secrete procoagulates, and this is to help clot. And they secrete chemicals to attract neutrophils. I'll just say just to attract other white blood cells. They can help destroy bacteria. And they secrete a growth factor for tissue repair. Okay, so those are some of the very important function of platelets. The lifespan, they live about 5 to 6 days. And a couple of terms, thrombosis. This is abnormal blood clotting. And an embolism is a abnormal blood clot that can travel. If it travels to the brain, then you can have like an aneurysm, a stroke. All right, so just quick review. We have hemostasis, cessation of bleeding. Platelets are fragments of the megakaryocytes, which are from the cell's monocytes. Why do they get so big? DNA duplicates without cell division, so they cannot get out of the bone. So you can kind of think as the bone. So, so if this happened to be, you know, in a bone, the cell can't get out, but its tendrils can. It gets in the blood supply and the platelets let loose. I've listed the function of the platelets, lifespan, what it means, thrombosis, and embolism. Last little bit, and we're home free. And this is hemostasis. Remember, this is the cessation of bleeding. How do we stop it? Let me get that a little bit better. So, sorry, there was a little step away for a second. I'm back. So we're going to talk a little bit about the three mechanisms of hemostasis, cessation of bleeding. There's three mechanisms. One, vascular spasm. Two, platelet plug formation. And last, blood clotting. So for vascular spasm, this is, I need to do a little scratch paper here. So normally, you know, you have your blood vessel, all right, and the red blood cells, white blood cells are running through here, all happy, big nucleus there. They're flowing through great, all right? So, but then let's say that all of a sudden you got a splinter, okay? So you've injured the tissue. As soon as that tissue is injured, it's going to cause the platelets to stick to it. So you can actually remove the splinter, but you still are left with an injury. Prior to the injury, there's something that is lining these blood vessels to help the cells flow real freely through it. And what this lining is called is prostacyclin. Okay, and this, I don't know if you've ever fried an egg in a fry pan. The fry pans today, they're not cast iron, they're coated with Teflon. And that Teflon causes you don't have to use a whole lot of oil to fry your eggs in. Well, it's kind of like the Teflon, prostacyclin is like a Teflon inside your blood vessels. It causes the cells to slip through easily. But as soon as the injury happens, then collagen fibers are exposed. Collagen, you should remember that from connective tissue. And what happens, platelets stick to it, okay? So back here, let's first look at this. So with vasospasm, injured tissue causes the platelets to release serotonin, which causes vasoconstriction. Okay, remember why? Because you don't want to slow the bleeding. Okay, and this is one happens until numbers 2 and 3 can kick in. The next 2 after that, you have platelet plug formation. And with this, first of all, you need to remember that platelets will not stick to healthy tissue. However, if there's an injury, healthy tissue due to what I just told you, prostacyclin. Okay. But when injured, the collagen fibers of the endothelium are exposed, which attracts the platelets. And I think I'll post a video to show this a little more as well, to attract the platelets. Okay. So then the platelets kind of have like pseudopods, they kind of makes like a spiny pseudopod that can actually help contract the tissues back together. So this is the start of a clot. And this is why you're very delicate. You should really just blot and not wipe an injury, because every time you wipe, you're breaking down these collagen fibers platelet plug that's trying to get started. The last phase is coagulation of the blood. And this is when you take fibrinogen, which is found in the plasma, and convert it to fibrin threads. And this is actually the clot. Normally I would go into a cascade of pathways for this. This right here is very involved to go from here to here, from fibrinogen to fibrin. And I'm not going to go into detail here. So it's like 30 reactions, and it's a huge cascade, but you do not need to know that. If you know that fibrinogen is converted into fibrin, and thrombin is responsible, as well as other things, that's an enzyme, then that's how you get the fibrin to thread. So I'm really going skimpy on this area. You can read over it, but like I say, I'm not going to hold you to this part. I will say that over time, the clot must dissipate. Over time, the clot needs to dissolve because the injury's healed, and plasmin is the enzyme that will do this. Okay, so I'm sure please start answering the review questions. That will help you in preparation for the exam. All right, guys. Don't even like doing this into the dark, but not having people out there to talk to. But anyway, hopefully you've learned from this, and I'll see you at our next lecture. Bye-bye.